Hello everyone and welcome to today's EAStalk webinar, part of our ongoing series brought to you by the European Aquaculture Society. My name is Anna Veskovic and I'm the project and the program manager at EAS. I will be your host for today's session. For those of you that are new to the EAStalk webinars, this series aims to present a broad range of topics that reflects the multidisciplinary nature of aquaculture, with insights from across our network, including researchers, students, industry professionals, and EAS corporate members. Today's session will last for 45 minutes with a 30 minutes presentation, followed by a 15 minutes Q&A. Please note that participants will be able to ask questions in the chat, and we will address as many as possible during the live discussion at the end. As always, the webinar is being recorded. It will be available on the EAS YouTube channel for 1 month, after which it will be accessible only to the EAS members on our website in the EAStalk Webinars archive. If you are not yet a member, I encourage you to check out our membership options. Free membership is available for students and retirees, giving you access to all EAStalk materials going back to 2019. Now let's move on to today's presentation, enhancing mucosal robustness in fish using yeast cell wall supplementation. In this session, we will explore how in feed supplementation with distinct yeast cell wall products can strengthen mucosal barriers, particularly the skin mucus across several fish species. This research highlights how yeast cell wall products can improve both the quality and quantity of mucus, enhance antimicrobial activity, and accelerate wound healing, even under stressful conditions such as pathogen exposure, challenging environments, or common aquaculture management practices. These findings have significant implications for sustainable fish health management, especially in salmonids, Mediterranean species, and widely used model fish. We are pleased to welcome Dr. Camille Houdelet, who will guide us through these insights. Dr. Houdelet is a research and development project leader in aquaculture at LALLEMAND, one of our EAS corporate members. She brings a strong background in fish health and stress physiology, having spent 4 years as a postdoctoral research at INRAe and Ifremer before joining LALLEMAND. Her work bridges fundamental research and applied innovation, contributing to the development of functional feeds that promote resilience and well-being in aquaculture species. As I said, LALLEMAND is a corporate member of the EAS, and a part of their membership benefits they are presenting the webinar today. And LALLEMAND is a yeast bacteria producer. So without a further delay, please join me in welcoming Dr. Camille Houdelet. Camille, the floor is yours.
Thank you, Anna. Good afternoon, everyone, and thank you to be there for this EAStalk. Yes, so today I will present you a selection of some trials that we generated in l'Allemand Animal Nutrition R&D team, and with a specific focus on fish mucus and skin health improvement with the yeast cell wall products. First, let me introduce to you a little bit the l'Allemand group. So l'Allemand was founded in Montreal by Fred l'Allemand at the end of the 19th century. And since 1923, we are specialized in yeast production. With over 5,000 employees globally, on the l'Allemand Animal Nutrition Department, we are around 500 people and present globally worldwide. If we focus on LAN Aqua, I will give you some key figures. We are working on more than 15 fish and shrimp species for our studies. We conducted around 15 trials each year on target species that conducted to more than 90 peer-reviewed papers in scientific publication or for commercial supporting. We are also the owner of the first probiotic authorized in Europe for use in fish and shrimp. And one of our big strengths is internal research scientists specialized in microbiology and fish and shrimp health and nutrition. And we created an active network of research partners and startups across the globe. We also carry a bank of marine organisms. For today, the plan is this one. So I will give you some generalities, and after I will describe some selected trials in different conditions, non-challenging conditions, handling, environmental stress or pathogen exposure, and I will finish with a take-home message. Let's start with the generalities. So the intensive farming results in really various repetitive insults for the fish. In fact, the fish are continuously exposed to various stress. It could be pathogen with bacteria, parasite, various non-infectious agents, but also submit to environmental changes and environmental stress like transfer, handling, treatment, net cleaning, and also changes in temperature. These stresses are applied in the various stage of life of fish, and it will conduct to a risk of develop complex pathologies, and these complex pathologies will leading to a decrease of the production performance and a poor health of fish. In LALLEMAND, we are working on preventive health and functional nutrition to manage the risk to these complex pathologies. And here I will just present you this graph. So you have to imagine three categories, three zones. You have the health zone. Fish are good with a good immunity, good performance, and a low level or no stress level and no pathogen. The second zone is a risk zone. You have the presence of some pathogen or stress events, and your immunity is a little bit tested. And if you go in the third zone, this is a disease zone. We don't want to go in this zone because the pathogen load is really too high compared to the immunity, and you will lose performances at this stage. So with the prevention strategy of LALLEMAND and using the functional nutrition, we really aim to mitigate the level of going in the risk zone and stay in the health zone to maintain the fish in good health. Today I will focus on skin mucus, but why? The first reason is because skin and the associated mucus layer are the first line of defense for fish. Not only a physical barrier, the skin and mucus associated are really involved in the osmotic balance of fish. The mucus is really dynamic and responsive to the environmental changes. And the properties are that mucus is really rich in bioactive molecules. You can find antimicrobial peptides, lysozymes, lectins, immunoglobulins, and proteases. And more, we observe that research try to work on mucus as a substrate to identify biomarkers of health status, because this is non-invasive substrates. So we observed a lot of more and more studies to evaluate the potential of the mucus to develop some tools to follow the health status. And what is really interesting for us, it's the functional feed ingredients can enhance the mucus composition and stimulate the immune functions. In LALLEMAND company, we are mainly worked on a yeast cell wall. And why the wall part is really important, because in the wall part you have two layers with two major molecules, the mannanoligosaccharides and beta-glucans, and these molecules act really differently on the immune systems. If you look at mannanoligosaccharides, so MOS, They are present in fish and microbes, and that it will be detected by the fish as a signal of a self-damage, like a physical integrity warning. It will induce mucosal regeneration. In the other part, the beta-glucans, they are present only in microbes. So if fish detects the beta-glucans, it will signal a potential pathogen, like a pathogen warning. And in this case, it will stimulate the antimicrobial response of the fish. But during all the presentation, you have to keep in mind that all the yeast cell walls are not the same. We proceed to strain selection and origin to obtain different yeast products. We optimize the production process also, and that leads to different MOS and glucans characteristics. It could be the quantity of the ratio of the MOS and glucans. It could be also the length of the chain, the bioactivity. And with these characteristics, the interaction with the receptors in fish will not be the same. So you will stimulate various receptors and leading to different immune response in fish. That is for the general context, and I will present you the general materials and methods that we used for our trials. The first thing is that we are working with multiple species. It could be Mediterranean species, salmonids, and model fish like zebrafish. We are doing trials in a controlled environment with temperature and salinity control, and we try to have quadruplicate tanks and at least triplicates. For the feeding procedure, we are using the control diet. It's a basal diet according to the species demand. And for the yeast cell wall application, we could use the pre-extrusion process or top-coated. The classical workflow is the following. You have a period of acclimation for fish, and this acclimation is followed by 5 to 7 weeks of nutritional preparation with control feed or yeast cell wall application in feed. This period, after this period, we applied challenge. This challenge, it could be acute stress, it could be chronic challenge. So it really depends of the challenge that we want to test. So you have to consider that it could be during several days or only during 1 or 2 hours. And when it's pathogens, it will be a bath challenge. After this challenge period, we continue the feeding with the yeast or the control feed. And it will be during 2 to 5 weeks post-challenge. During all the period of the trial, we will select some we will collect some samples at pre-challenge, at different time points during or after the challenge, and at the end of the trial. We sampled different tissues, so tissue sampling that I will detail you just after, and the macroscopic scoring in case of pathogen screening. For the general sampling, so as I mentioned, for the macroscopic scoring, for example, when we tried some Tennessee tests or AGD, we will observe some lesions on mouth, gill, or skin. So we will use some classical table to evaluate the score of the disease. And for the tissues, we classically sampled gut, gill, skin, mucus, and blood to do a different kind of analysis that I detailed here. And for the mucus, most part of time we are doing some biochemistry. We also analyze the physical properties with viscosity and the gene expression. I will detail a little bit more for the mucus collection because this is the topic of today. For the previous method that we used to collect the mucus, we used the wipe method. So it consists just to wipe the fish, but it's not really consistent because you have a high variability between fish. So it's not really relevant to evaluate the mucus quantity, the mucus excretion, to compare between your condition and between your fish. It's not really the best, but it's good because you can do the biochemical analysis on this. For the new method, so today we are using these methods for the quantity, to evaluate the quantity. This is a sponge strip. So for that, we are using a specific sponge with known size and weight, and we will put the sponge during 30 seconds on the one flank of the fish, and after we will wait to obtain a quantity of mucus per square centimeter of fish. This is more standardized, so it's easier to compare the mucus excretion. But with this method, you only have the quantity and you cannot obtain the quality of the mucus. So you still have to use the other flank of the fish to sample the mucus. This is for the generalities, and now I selected some interesting trials, I think, in various conditions. First, let's start with the non-challenging period. The objective of these first trials was to evaluate the effect of a yeast cell wall product at 2 doses on the sea bream and on the quality of the sea bream mucus after 10 weeks of nutritional preparation. For the mucus quantity, we clearly observed an increase of the quantity of mucus with the 2 yeast cell wall products, and this mucus quantity was correlated with the concentration, the protein concentration. We also looked at the gene expression of mucins. So mucins are the major components of mucus, and we observed a significant increase for the 3 genes selected here. We observed an increase of the expression of these genes that are involved in the cellular adhesion and immune function of the mucins. So we clearly observed the beneficial effects of the product at the highest dose on the mucus properties. Another non-challenging trial, this one on the Atlantic salmon, and in this case we tested 2 combinations of yeast cell wall. This time it's also a little bit different compared to the other trial because we analyze the viscosity, so more the physical properties of mucus. Here in gray, you have the control mucus with the high initial viscosity and a moderate response to shear. The second yeast cell wall product, you have a strong structuring under shear due to the slope that you observed on this graph, and it's meaning an increased barrier effects. For the third product, the viscosity is low but stable, and it could be more favorable to regeneration. So for this trial and the other one, we have interesting parameters on mucus that are improved by different yeast products. If we go now on handling, I selected this trial because it was interesting to we tried to mimic the kind of lesions that you can observe in transfer, for example, in tongue transfer of fish. So I mentioned as mechanical wounds. For this trial, we used the zebrafish model, and we tested 2 diets, 2 experimental diets, the beta-glucan, known to be involved in the wound healing, and multi-strain yeast fractions. So we applied the biopsy punch on the flank of the fish of a known diameter, and after we observed the evolution of this lesion over time. If you look at the beginning of the time, in gray you have an increase of the healing profile not of the healing profile. This is of the wand. You have in the beginning you have an increase of the wand, but in the 2 experimental diets you already go in the healing profile. We don't really see the difference after, but it's more visual, maybe on picture. For example, if you look at the 8-day post-wand, you observe that the size of the wand is reduced with the 2 yeast products. But this is not really a good quantitative method, so we also did some histology to analyze the granulation of the tissue. So this is the granulation at 4 days post-wand, and we observe the area of the granulation tissue. And we clearly observed that the multistrain yeast fraction was really better for the regeneration of tissues and really help for wound healing. If you want more information, this study was already published in Aquaculture reports. Another handling stress, this time it was netting and the high density stress during 20 days. And we applied this stress to the European sea bass, and we tested 3 doses of yeast cell wall. The first result is the mucus excretion. If you look at the stress period, at the end of the 20 days, we observed that the highest dose of yeast cell wall, we observed the higher mucus excretion. So we observed the beneficial effect of the yeast. And for this trial, I will just present you some other results, not directly related to the mucus, but more to the gut, because we tried the fecal calprotectin biomarkers to evaluate the beneficial effect of our product. If you look at the first period, in the non-challenge period, the fecal calprotectin was really higher at the higher dose. So it seems that the fish detected the yeast cell wall pattern, and we have activation of local immunity. If you look at the fecal mucids in parallel, in the non-challenge condition, you already have an increase of the mucins that mean reinforcement of the gut function barrier. If you look at the stress condition, you have a decrease of the fecal calprotectin and still continue to increase the mucins, meaning that we saw that there is a resilience to the stressors due to the diet, to the yeast cell wall diets, and we suggest that we have a lower stress induced due to the presence of experimental diets. So this study was recently submitted in Fish Physiology and Biochemistry. And for me, it was really interesting to show you biomarkers that are new and that are directly correlated to the fish health. For the environmental stress now, I will present you this trial with the objective of this one was to test 3 doses of yeast cell wall, but in condition of a thermal stress. We applied 4 degrees more than usual temperature for Atlantic salmon. We observed that the statistics didn't show a significant increase of the crude skin mucus, but if we look at correlation between the skin mucus production and the moss, we observed a positive correlation. So when you have more moss in your diet, you produce more skin mucus. We also analyzed the properties of this mucus, And we tested this mucus in vitro against the Tennessee-Baculum maritimum. And we observed a reduction of growth of the pathogen in the yeast cell wall conditions. With this trial, we show that we have a clear beneficial effect of the moss supplementation in the fish mucus production and qualities. For the pathogen exposure, I will go back to one study that I presented you just previously, but I presented only the pre-challenge part, and now this is the post-challenge part. So this is a study on Atlantic salmon, and we tested two combinations of yeast cell wall. After a nutritional preparation, we applied amoeba gill disease challenge. In fact, it was a repetitive stress of a repetitive bath challenge. And we observed really interesting results on gill score. So for the amoeba, we are working on gill because amoeba are visible on arc of gill arc with the white spots. So we can use tables to score this the propagation of amoeba. And in this study, with the two yeast cell wall products that you have here, you clearly decrease the score of the pathology. Seeing these results, we go to the mucus analysis. And if I show you the previous results pre-challenge, you were on this condition with the control here, the first yeast cell wall and the second here. After challenge, the pattern is really different. We observed that the control has a really decrease of a loss of structure with a very low viscosity. So the fish fed with the control lost the structure of the mucus. And for the two experimental diets, so for the first one, we observed a high viscosity and we maintained the protection between the pre-challenge and post-challenge conditions. With the second yeast cell wall product, we observed a moderate viscosity, maybe a balanced response of the mucus in these conditions. So these results were really interesting because it's more physical information. And we know that the mucus is really important for this biochemistry because of the molecules that you have inside, but also with the viscosity to retain or not the pathogen. So it was really interesting for us, and our product acts significantly on that. The last trial that I want to present you today is one on sea lice challenge. We tested one commercial diet and we tested one yeast cell wall product on Atlantic salmon. The first result is the crude mucus quantity. Pre-challenge, we observed an effect on the production of mucus in yeast cell wall conditions, and also 3 weeks after the challenge. So this is the first benefit that we observed. If you look at the lysozyme activity here of the mucus, so lysozyme or antibacterial enzyme that will help in the immune defense. If you look at these results, you have the 1 week, at 1 week post-challenge, we observe an increase of the lysozyme activity compared to the control. And what is interesting, when you look at the general number of sea lice that we counted on fish, here we observed that the fish with the highest mucus quantity before challenge were also the fish with the lowest quantity of sea lice on their body. So we clearly enhance the skin barrier prior, but also in response to the sea lice. This study was also published in Aquaculture, so if you want more details, do not hesitate. Now it's time to conclude. So today I presented you various T cell walls. So don't forget that we have different yeast cell wall products developed with different process, different strains, and different qualities. And all of these yeast cell wall products demonstrated a beneficial effect on the skin mucus properties that we analyzed in various conditions, ideal conditions, but also stressful conditions. And I didn't present everything on studies this time, but we don't look at only the skin mucus. We also observe performance of fish and other parameters to evaluate the global health of fish. And we clearly conclude that the yeast cell wall products that we are using are good preventive health tools in multiple contexts. With this expertise, we can help to choose the better products, but we have to keep in mind that we have to adapt to the context of the field of the fish. We have to take into account the seasonality of fish, the biological targets, also the duration and the dose that we can adapt depending of the objectives, and also to be in the good cost-effective strategy for the target application. This is for me the end of the presentation, and I really want to thank you for your attention. And if you have any question, do not hesitate. And if you are too shy, you can also contact me after by email. Thank you.
Thank you so much, Camille, for fascinating and informative presentation. I really enjoyed it, and amazing examples from LALLEMAND. I think it's quite clear that yeast cell wall supplementation has potential in promoting mucosal robustness, improving disease resistance, and enhancing overall fish welfare, especially under the challenging farming condition, which you just showed us. The data you shared across different species and stress models really highlights how adaptable and effective this functional feed strategy can be. We are now moving to the Q&A session. So this is your time and opportunity to ask any questions you might be having to Camille. So please type the questions in the chat box, and we will go through as many as possible in the time that we have left. So let's get started. I will start with the first question. What is the relation between feed and effect on mucus quantity and quality?
Yeah, because sometimes when you think about it, you will give some food to fish and you will observe the effect on the skin mucus. And in fact, there is different elements that support that observation. The first one is because yeast cell wall will interact with receptors in the guts, with immune receptors, and it will stimulate the global immunity of fish. And of course, these molecules are really active in the mucus, as I presented previously. And that's why we observe that. There is also some other clues on the gut skin axis and the role of microbiota, but we didn't really look at this part for now, but it's also an interesting axis, I think.
Thank you, Camille. I would like to encourage you to not be shy and to still ask a few questions for Camille. As she said that she left her email address on the last slide, so make sure that you mark it down in case you feel a little bit too shy to ask the questions, but please do not, as we really want to have really good discussion today. Camille, if you don't mind, I will ask my second question. For antibacterial effects of mucus, have you tested other bacteria?
Yes, for the trial with the in vitro test, we also tested Moritella and other subspecies of Tennessee but they are not really, they are not the more, we didn't observe more results or the same results. We observed inhibition, but it was not in the same pattern. It was the Tennessee baculum maritimum was really impressive for the skin mucus effects.
Thank you. We just received a question from Mustafia Saidu. Can I have a brief info on how to develop a biosecurity plan? Thank you.
To develop
a biosecurity plan.
For me, it's not clear, the biosecurity plan.
Okay, so
sorry.
Mustafa, can you please explain a little bit more the questions that you have for Camille? In the meantime, we received a question from Tornike Lasha Karashavili. In saprolenchia case, is good these additives?
We know that saprolinea is really a major problem in fish, and we are working on it, to be honest, because we have really interesting first results in our in vitro lab. So we hope to obtain really good. We have already good results in vitro, but we are still waiting for in vivo parts. But these additives are really adapted to this kind of pathologies. Because of this pathology, it's also skin pathology. So we are convinced that it will help to at least to display the effects.
Thank you. The next question we got is from Strö Maria Desmira. Have you done any testing on sturgeons?
I started 1 year ago in LALLEMAND company, so I'm not sure to have the information. Sorry, I cannot answer to this question because I'm really not sure, but it's not classical for us to use this fish species. But maybe we have one trial. I'm not sure of that. Sorry.
Thank you. No problem. Thank you. The next question we got is from Anna Papadopoulou. Thank you very much for the excellent talk. The fish mucus is important in fish immunity to defense pathogens, but also too much fish mucus in tanks might be good food nutrients, for example, bacterial pathogens on tank surfaces. Do you have any opinions about this?
Yeah, I completely agree with Anna. That's why we also looked at the physical properties, because you have if you have a good viscosity, for example, if you have a high viscosity, you will maintain the pathogen in the mucus, and you have two effects. You can have more development of the bacteria, or you can also let more time to the molecules to act against these bacteria. So it's really dependent of the ratio between the quality, the biochemistry quality, but also the physical quality of the mucus. That's why you don't have to consider only one part of the mucus. For me, it's a really complex matrix, and you have to look at different elements.
Thank you. The next question we got is from Sarah Solli Knutshaug. Hi, Sarah here, PhD student at Nord University in Bodo in Norway. She has 2 questions. So the first question, Camilla, you can answer that, and then I can answer on the second question. So the question 1 is, have you observed any disadvantages of the yeast cell wall? If so, what?
In fact, if you think about the beta-glucans, for example, beta-glucans, for example, we don't advise to use it in continuous feeding phase, because as I presented, the beta-glucans are mimicking the pattern of microbes. So it's really danger signal. So if you have this danger signal in a long period, you will create immune fatigue, immune depression. So it's really not favorable for fish. That's why it's really important to organize the strategy of the feeding depending of the pathogens that you want to prevent, or if it is in a specific period. So yes, you have to really to design well the strategy.
Thank you, Camille. So the second question from Sarah was, is it possible to send the articles mentioned in the presentation in the chat? Would love to read them. So what I wanted to say to Sarah is that I'm not sure if you're a member of the EAS or not, but since you're PhD students, you can join the EAS for free. And after this webinar, we will publish Camille's presentation on the EAS website. So you will be able to get the presentation slide and to see a little bit more about the articles that are published. But maybe Camille can as well say to Sarah if you're able to send the articles directly to her or not.
Yes.
So Sarah, please email Camille. You have her email address here on the screen. The next question we got is from Tornike Lascaravascavili. I'm sorry, Tornike. Is Fibocell good for the skin?
To be honest, because I am here only since 1 year, I didn't have the opportunity to work on data on Fibocell. So I will not go on this subject because I'm not really comfortable on this project. So because we don't have a lot of study on this one, and not specifically for the mucus promotion.
Thank you, Camille. The next question we got is from Bia Pinko. Hi, Camille. Thank you for your presentation. I don't know if I understood it right, but when using the new method for mucus collection, you said that you also use the wipe method, but on the other flank of the fish. So what are the different analyses that you do using each method?
Yeah, maybe I can just go back on this. Maybe not. Yes, I can. Yes, we are using two methods depending of what we want to observe. But as I said before, it's important to analyze the quantity, the quality, and different parameters of quality. I'm looking for this. Okay. In fact, I said previous method and new method. But for the quantity, we are using this method to be really reproducible. But we are still using a wipe method to sample the mucus just for biochemistry analysis. For example, we want to evaluate the lysozyme inside or the antibacterial effect in vitro. We have to collect the liquid. And with the sponge method, we don't have the liquid. We absorb the mucus on the sponge, and we just weight the sponge. So that's why we have to use the two methods. But for quantity, to evaluate well the quantity, we have to use the sponge strips that is more reproducible between the fish.
Thank you, Camille. The next question we got is from Tom McDermott. Any work on the salmonid Rixial septicamai?
I will say the same thing that previously. This year we didn't test this pathogen, but maybe in the previous year, but I'm not sure. And because I'm too young on the company. No, I don't have the data on it, but I think my colleague is in the staff, but but I don't have the answer on this pathogen.
Thank you, Camille. And in the meantime, maybe Eric can write something in the
Yes.
chat as well, because we know he is here. The next question we got is from Rashid. Thanks for the nice explanation. How about fish lose like Argulus parasite?
Sorry, it will be the same answer. I don't know. I'm not familiar with this one. This is, I don't know. I'm really sorry. If Eric is here, maybe you can answer to these two questions if we already tested these two pathogen challenge.
Thank you, Camille, and thanks to Eric as well. So we have a few more minutes for the Q&A. So please type your questions since until we don't wrap it up for today. The next question I will be having for you, Camilla, is why the multi-strains yeast fraction is more effective for mechanical wound healing?
For the, I will show the slide. Maybe it's better to explain. So this is for this trial of mechanical wounds. The multi-strain yeast fraction, we selected Saccharomyces and non-Saccharomyces yeast for this product. And the really big interest to do that, it's to stimulate different receptors in the same way. So you will go in different receptors. So you will activate different immune pathways, and like this, you have a response, immune response that is more effective.
Thank you. And what about which yeast technologies can induce immune fatigue?
This is I mentioned previously that, for example, the beta-glucans we have to be really careful with the usage of beta-glucan. It's really punctual usage, for example, and not in continuous feeding way.
Thank you. Just a last reminder for all of you that we have 1 more minute left. So if there is any question still for Camille, please type it down. So Camille, do you recommend different in-feed doses for different seasons, hence feed intake?
For this question, yeah, for example, in the winter, we can think to use different doses, but in fact, with the yeast cell wall, we don't need to reduce the dose or to increase the dose, because you have the feed intake is lower during this period, and the yeast cell wall will stay longer time in the gut, and because the transit is slowed down, so you will have you will maintain the effect of the yeast cell wall longer also in the gut. So this is not necessary, but as I always said, we clearly have to adapt to the conditions of the fields and of the fish, and we can add more trials to refine maybe the dose depending of the seasonality, of course.
Mm-hmm. Thank you so much. Since we did not receive any more questions, I would like to wrap up today's webinar. So thank you again, Camille, for sharing your expertise and for giving such a clear look into the science and practical value of functional feeds using the yeast cell wall components in aquaculture. For sure, today's session highlighted how innovative feed solution backed by research and field trials can support more sustainable and resilient aquaculture practices. We would like to thank LALLEMAND, one of our corporate members, for supporting this presentation and for their ongoing contribution to advancing fish health and nutrition in the industry. As a reminder, this webinar was recorded and will be available on the EAS YouTube channel. I will be sending you an email with the link of today's webinar if you would like to watch it once again. Thanks again for joining us today, and we hope to see you at one of our next YCW webinar or upcoming events. In the meantime, we got one last question. Do you mind, Camille, to answer to that one?
Okay, let's go.
So it's from Stroe Maria de Smira. Thank you for interesting presentation. Are they commercial fish feed that already include yeast cell wall?
Yes. Yeah. There is some commercial fish feed with yeast cell wall product. We are producer of yeast cell wall, and we are already distributed our product in feed producer company. So it's already applied.
Thank you, Camilla. I would like to thank you all for joining us today and looking forward to see you on our next EAStalk webinar.
Thank you.
Thank you.